Okay, so this is chapter 12, part 2, and we're going to continue our talk about Mendelian genetics and heredity. We left off talking about the 3-to-1 ratio, and we're going to get back to that in this part of the chapter. But first I want to define some terms for you. And so I want to make sure we understand the difference between a dominant allele and a recessive allele. Okay, so we say individuals either express the trait in a dominant fashion or a recessive fashion. So dominant traits or dominant alleles are those that are always going to be expressed. Okay, whether the organism is, we say, true breeding, which we can also use the term homozygous, or hybrid. We can also use the term heterozygous. So for example, if we think back to what we left off with, looking at flower color in pea plants, purple or violet flower color, we would say is dominant to white. And so if we looked at the parental generation, right, we've got true breeding, homozygous, dominant, purple flowered plants, and also the result of that first cross, so the F1 offspring, the hybrids, were also purple, even though they are going to be considered heterozygous. Okay, so the dominant allele always gets expressed. Recessive alleles or recessive traits are not always expressed. In the heterozygote, or again, hybrid organism, we say that recessive alleles are masked. Okay, so for example, again, if we look at the F1 individuals from that last cross that we looked at, which we're going to look at again, the F1 individuals, the hybrids, the heterozygotes, they were purple, not white. Okay, so the white allele, the allele coding for white flowers, was hidden in the heterozygote. The only way to get a a recessive trait to show up is in the homozygous recessive individuals. Okay, so we'll come back to that again. The key to remember, as we saw in Mendel's cross with his pea plants, is that the recessive trait, so white flower color, isn't lost. It's not absorbed in the hybrid, because remember, in the F2 generation, it reappeared. Okay, so again, more evidence that the blending hypothesis is not true. Okay, so what else did Mendel tell us? So Mendel proposed that plants inherit 2 copies of a trait, which we would call alleles now, right? He didn't have that term, but he said organisms inherit 2 discrete units, which we would call alleles, for that particular trait that he was studying first, flower color. He said that diploid organisms inherit 2 of those discrete factors, which again we call alleles now, for each gene, right? So all diploid organisms, including organisms like us, humans, and corn, which we'll study in the lab, are diploid. So we inherit 2 alleles for every gene. And you have to think back to meiosis for this. So each parent involved in a cross is going to transmit one of its 2 copies of the gene to its offspring during fertilization. So again, you got to think back to meiosis, make sure you understand it, make sure you understand that gametes are haploid, they carry 1 allele. 2 haploid gametes come together during fertilization to produce that diploid zygote, which again is going to have 2 alleles for each gene, 1 from each parent. Okay, so let's look back at our flower example. Okay, so this should look familiar. We looked at this back in the first part of the chapter. So we have our parental generation, our F1 generation, and our F2 generation. Okay, so we have 2 crosses here. So again, we're looking at our trait, which is flower color, and we know now that the dominant allele is purple and the recessive allele is white. Okay, so let's go back up to our parents. We would say here, this parental generation, remember we use the term true breeding or pure lines, so we know that these are homozygous. They have 2 of the same alleles. So the violet or purple plants here would have 2 purple alleles. The white plants would have 2 white alleles because, again, they're homozygous, so they carry 2 of the same allele. When we cross these, okay, 1 allele from each ends up in the gamete, and then those gametes will come together. Well, the violet plant only has the purple allele to give, and so that's what it's going to give. The white flowered plant only has white allele, so that's what it's going to give. So all of the offspring of this cross will end up as hybrids or heterozygotes. So they'll have one purple and one white allele, so they have two different alleles. Okay, when we cross these, okay, so again, two heterozygotes being crossed, this is where we get that 3-to-1 ratio. Okay, so for every white flowered plant, we will have 3 times as many purple flower plants. Okay, this is also how we know that purple is dominant to white. Okay, so let's make a little bit more sense of this. So when we talk about purple and white, this physical expression of a trait, what we actually see, that's going to be called the phenotype. Okay, so that's a term that you want to make sure that you know. So again, purple flowers, that's the phenotype. The white flowered plants, phenotype is white flowers. If we're looking at human traits, having dimples, that's a phenotype. Not having dimples is the alternate phenotype. Okay, so again, it's a physical expression of a trait. Genotype, on the other hand, are the actual alleles that an organism has. So it's the organism's genetic makeup. How do we demonstrate alleles? By using either 1 or 2 letters. Okay, we always use a capital letter to represent the dominant allele, and a recessive allele is represented by a lowercase letter. So here's some nice examples. So when we talk about flower color, the purple allele would be represented by a capital P, and the white allele would be represented by a lowercase p. So that tells us purple is dominant to white. In lab, you're going to study a couple of different traits or characters. One of them will be kernel color. And so purple kernels are dominant to yellow kernels. Okay, so the phenotype would be purple kernel. The allele for purple kernels would be capital R. And the allele that we're going to use for yellow kernels will be lowercase r. You'll also be looking at kernel shape. So you're going to hear starchy, which also means smooth. That's dominant. We're going to use a capital letter S. And then sweet, which is recessive, which is also wrinkled, we will use a lowercase s to represent those alleles. Now for genotypes, if we have a white flowered plant, the genotype for that plant would be little p, little p. So we have to have 2 alleles because it's diploid, and both of them would have to be recessive in order to get the recessive phenotype. A purple flowered plant could be either big P, or big P, little p. So both the homozygous dominant and heterozygous genotype would give us the same phenotype of purple flowers. Okay, on the next slide I have some more examples. Okay, so again, if we're talking about some stuff that we did in lab, this first one, purple kernels could be big R, homozygous dominant. Okay, so everything listed here, sorry, on this slide, let me back up. Everything listed here on this slide is going to be a homozygous genotype and a homozygous phenotype. Okay, in the homozygous condition, the organism has inherited 2 of the same alleles for a specific trait. So again, in lab, when we look at kernel color, one phenotype that you will see are purple kernels, and one potential genotype would be big R. We would call that the homozygous dominant genotype for corn kernel color. If we're talking about Mendel's flowers, big P would be the homozygous dominant genotype for purple colored flowers. Again, back to lab, if we're talking about yellow kernels on our corn cob, little r would be homozygous recessive. Those kernels would be yellow. Back to Mendel's studies, white flowers would be coded for by little p, homozygous recessive genotype. Okay, so again, everything listed here is homozygous. The letters here, big R, big P, little r, little p, those are genotypes, right? That's the actual alleles inherited. The phenotype is the physical expression. So purple kernels, purple flowers, yellow kernels, white flowers. Those are all phenotypes. So you want to make sure you know the difference between a genotype and a phenotype. Again, these are all homozygous. On the next slide, I have some more examples of heterozygous. So you can also get purple kernels from this genotype. So capital R, lowercase r. So again, those are the two alleles here, and the two alleles are different. Okay, so this individual inherited both a dominant and a recessive allele. And whenever that happens, if it follows Mendel's rules, then the organism will express the dominant phenotype. Okay, so big R, little r would give us purple kernels. Big P, little p would give us purple flowers. Okay, so again, you want to make sure you know the difference between genotype and phenotype, and the difference between homozygous and heterozygous. Okay, so let's do another example of a monohybrid cross. Okay, so again, the flower color that we've already looked at, right, the parental, the F1, the F2, that was another monohybrid cross. Here's another example here, looking at corn kernel, seed color in flowering plants. In lab, you will look at corn kernel color. What is a monohybrid cross? So when you hear the term monohybrid cross, this is where you are following the inheritance pattern of a single gene. Okay. We'll also talk about dihybrid crosses in the next part of the chapter. In a dihybrid cross, you're looking at the inheritance pattern of two genes at the same time. So when you hear mono, think one gene. So you're following the transmission of one gene. We can do this by using something called a Punnett square. And so I'm going to walk you through this picture here on the left. So in this particular example, it's a monohybrid cross where we're looking at the inheritance or the transmission of seed color in pea plants. Okay, so just one trait, one gene I'm looking at, the seed color gene. The dominant phenotype is yellow, and the recessive phenotype is green. And so yellow seeds can either be homozygous, dominant, big Y, or heterozygous, big Y, little y. The only way to get green seeds is in the homozygous recessive condition, little y. Okay, so let's start up here at the top. So here is the parents. These are going to be both true breeding plants. So this particular plant is true breeding for yellow. Okay, so when you hear true breeding or pure, you're thinking homozygous. So in this case, this plant is going to be homozygous dominant for color, or big Y would be the genotype. And again, the phenotype is yellow seeds. This parent, on the other hand, is also true breeding or pure, still homozygous, but on this side, we're going to have homozygous recessive. And so again, the phenotype will be green seeds. The genotype would be little y. Okay? And so if I come back to this parent, the only type of gamete that this parent can produce would be gametes that hold the capital letter Y, the dominant allele, because that's all that this parent has. Okay, so all of the gametes, 100% of them, will be big Y. The same thing holds true for this parent. All of the gametes produced by this parent will hold little y, because that's the only allele present in this organism. So again, all of the gametes are little y. Well, when a gamete containing big Y and a gamete containing little y come together, they will form heterozygotes. Okay, so all of the offspring from this cross will be big Y, little y. That would be their genotype. Their phenotype is that they would all produce yellow seeds. Okay, so that hopefully makes sense so far. Now, if I were to allow two F1 offspring to mate, so that cross would be big Y, little y, crossed with big Y, little y. Let me actually write that so we see that. Okay, so again, it would be big Y, little y, being crossed with, I like to make an X to represent the cross, another organism that is also big Y, little y. Okay, so above I would put a cross in between these to show that this is a mating. Okay, and also if you notice, my capital letter Y and lowercase letter y look really similar. So in any case where the capital and lowercase letter look the same, so Y, W, P. Okay, what I usually do when I'm writing to make sure I don't make a mistake is I will take and I will draw a line above the lowercase letter just to help me with my notation. Okay, I know that that's a recessive allele. Okay, so now if I have this cross, right, so two heterozygotes from the F1 generation that are allowed to mate. Okay, so what kind of gametes can each of these parents produce? And so that's shown here. This genotype can produce gametes that either hold a capital letter Y or a lowercase letter y. And so again, you have to kind of think back to meiosis. Remember how that happens. And I'll show you that at the very end of this part of the chapter. But these will come apart during meiosis. And so half of the gametes, 1/2, will contain a capital letter Y, and the other half of the gametes, an equal number, will also contain a lowercase letter y. So 50-50, 50% of the gametes will carry the dominant allele, and 50% of the gametes will carry the recessive allele. And that's true for both of these parents, both this parent as well as this parent. And so here is the Punnett square. So what I'm going to do is I'm going to take the gametes of one parent and put it over here, and the gametes from my second parent and put them over here. So if this is parent number 1, and this is parent number 2, parent number 1's gametes are listed here on the left-hand side of the square, and parent number 2's gametes are listed across the top. Okay, and so again, for parent number 1, half the gametes are big Y and half are little y. And then for parent number 2, same thing, half of the gametes are capital Y and half are lowercase y. And so we use the Punnett square to demonstrate potential offspring. Okay, and so let's do it. So in this first box, this is saying if gamete 1 from parent 1 is fertilized by this first gamete of parent 2, we will end up with offspring that are big Y, homozygous dominant. If I come over here, this is saying if this gamete here from parent 1, which holds the dominant allele, is fertilized by this gamete over here, which holds the recessive allele, we end up with this genotype. This is a heterozygote, capital Y, lowercase y. The phenotype would still be yellow because yellow is dominant to green. Down here in this box, we've got this gamete being fertilized by this gamete. And so that ends up here. That's going to be a big Y and a little y. Notice how the dominant allele is always written before the recessive if both are there. The phenotype would be yellow. And in this last box, we have this gamete, coming together with this gamete. And so that would give us little y, homozygous recessive, green seeds. Okay, and so from this, we can see that 3 of the 4, 3/4, will be yellow. And 1/4, 1/4 will be green. So that's that 3-to-1 ratio. Okay, again, you're going to see that here in this lecture. We'll do it in lecture and class together. You'll also see that ratio in the lab. And so whenever you get a 3-to-1 ratio, keep pushing the wrong button. Whenever you get, that 3-to-1 ratio, okay, just like we did here, that indicates that it's a cross between 2 heterozygote organisms. Okay? Okay. So let's do one. So again, the Punnett square is a way to make a prediction about the possible outcomes of a genetic cross or a mating and their respected or expected frequencies. Okay, so here's my cross down here. So big R, little r, crossed with big R, little r. So 2 heterozygotes again. So take a minute, pause your video, and try to figure this out. Okay, so hopefully you pause the video. We'll come back together and we'll do this together. So I'm going to have, parent 1. I'm going to list the alleles or the gametes for this parent over here on the left-hand side. So half of that parent's gametes will hold the capital letter R or the dominant allele. The other half of that parent's gametes will hold or carry the recessive allele, lowercase letter r. I will then take parent number 2 involved in this cross and list its gametes on the top. And in this case, it's the same thing. Half of this parent's gametes will hold the dominant allele, and half will have the recessive allele. Okay, so inside the box, I'm choosing a different color, I'm going to put the offspring. And so in this first box, it's the product, right, of this gamete fusing with this gamete. And so I would end up with 2 dominant alleles. Okay, and if we're talking about, corn kernel color, this would be a purple kernel. In this second box, it's this gamete fusing with this gamete. And so that would be a big R, little r. And I would call this a heterozygote. It would still give me a purple kernel. Here, it's this gamete fusing with this gamete. So again, big R, little r. And again, that's going to be a heterozygote, still a purple kernel. And my last box here is going to be little r, homozygous recessive, and that would give me a yellow kernel. And so I would end up with a 3 to 1 phenotypic ratio. So again, for every yellow kernel, I have 3 purple ones. It's that 3 to 1 ratio. Again, look at the cross, the parents of the cross, 2 heterozygotes. Okay. So on to there we go. On to the next slide. I want to introduce you to something called the test cross. And this is going to be done as a mechanism to determine whether an organism that has the dominant phenotype is a heterozygote or a homozygote. So in the first case up here, I have a yellow seed, right? So a plant that produces yellow seeds. I know that it produces yellow seeds, but I'm not sure if the organism is homozygous dominant or heterozygous for that particular trait. And so I can figure out the genotype of that organism, the unknown genotype, by performing what's called a test cross. And so what I'm going to do in a test cross is I'm going to mate this organism with an organism that is homozygous recessive for the trait. And so in the case of peas, it would be a homozygous recessive green seed or pea producing plant. Okay, and so you can look here at the possible results. So here's one possible result here, and here's a second possible result. And you kind of have to fill in the question mark. What would go there? And so think about that for a minute. Again, you can pause me if you need to. So again, over here, these are little y's. And so this is a big Y, so this makes sense. Big Y, little y gives us a heterozygote. Big Y, little y gives us a heterozygote. How would we get heterozygotes over here as well? Well, that unknown, that question mark, must be a capital letter Y. Okay? The only way to get all of the offspring yellow would be if this was also a dominant allele. Come down here, how would you get 50% of the offspring yellow and 50% of the offspring green, or a 1-to-1 ratio, well, this unknown here would have to be a lowercase letter y. So again, I'm going to do my y with a line over it. It would have to be the recessive trait. Okay? And so you're going to see that term test cross. And so you want to make sure when you see test cross, you think, I have to cross this organism with a homozygous recessive individual. Okay? That is how you perform a test cross. I also want you to make notice, let me just get rid of N. Okay, I want you to make notice of the 1-to-1 ratio. So down here, this is another ratio that you will see both in lecture and in lab. And so you want to make sure that in the case of a monohybrid cross, the 1-to-1 ratio is the result of, and actually let me get my pen back, just make sure we got this. Ooh. Okay, there we go. Let me just change the color to pink. Yeah, there we go. So the way to get a 1-to-1 ratio in this case would have to be a heterozygote. It's being slow. So a heterozygote being crossed with a homozygous recessive. So here's a little y and a little y. That is the only way to get the 1-to-1 ratio. And so again, you will see that ratio again. So you want to make sure you know how to get the 3-to-1 ratio. It's always 2 heterozygotes. And then the 1-to-1 ratio is a heterozygote being crossed with a homozygous recessive individual. Okay, so for our monohybrid cross, you really want to make sure that you know those two ratios. And this is our last slide. I just wanted to make sure that you understood how this was happening. So I referred back to meiosis, and so I wanted to show you this again. And so in this particular example, we are looking at pea plants, and we are looking at the trait which codes for height. Okay, and so if we start off in the beginning here, this is showing one parent. Okay, so this is meiosis in one parent, and it's showing a parent that is heterozygous for this particular trait. So the genotype of this parent is big T, little t, and so you can see those alleles here on these homologous chromosomes. So we're in metaphase 1 of meiosis 1. So DNA replication has already occurred, chromosomes have already formed tetrads, and the tetrads right now are lined up on the metaphase plate. So these chromosomes, these homologous chromosomes, are going to be pulled apart, right, during meiosis 1. Okay, so we end up with 2 cells, and so each has its own chromosome. There's no more homologous pairs, they're in separate cells. This cell carries 2 chromatids, 2 sisters, that both carry the big T allele. And this cell carries, again, 2 sisters, 1 chromosome, that carry the little t allele. Meiosis 2 happens, right? This cell splits into 2. Each of these gametes have the big T allele. This cell splits into 2. Each of these gametes have the little t allele. And so there's 4 gametes formed, but if you'll notice, these first 2 gametes are the same. And these second 2 gametes are the same. So we say that half of the gametes here will have the big T allele, and the other half of the gametes will have the little t allele. Okay? So I want you to quickly demonstrate a cross between 2 heterozygote individuals. And by now you might say, I don't even have to do the Punnett square, right? I already know the ratio. If you need to do the Punnett square, take a minute to do that. And you should find that the ratio in the end is 3-to-1. 3-to-1 tall to short. Okay? We will be getting a lot of practice doing Punnett squares in both lecture and lab. And so that concludes the second part of the chapter.